Hello everyone. Welcome to today's workshop on segmentation and CellProfiler, principles 2D and 3D. My name is Beth Simony. I run the image assay development team for Ann Carpenter's lab, also known as the imaging platform at the Bird Institute. Hope everyone is doing well today. Let's go ahead and dive right in. So a major concept in object identification and a major step in image analysis is figuring out how do you actually find the objects of your interest. It's the hardest part of an image analysis task. Here I have some fly nuclei. I can see, based on my expert knowledge, that there are two. You can probably see that as well. But how I train the computer to know whether or not there are two is the hardest problem in image analysis typically. We've gone through this a little bit in the introduction, but today I'd like to go through this in a lot more detail, show you a lot more of the examples in CellProfiler of different ways we can configure segmentation, how actually all of this gets set up, and then how you can adjust it so that even when you have a few special cases in your experiment, you're able to handle those just fine. Now the first step always in an object identification in classical image processing is a thresholding. We're going to distinguish the foreground from the background, and then we're going to split and merge objects properly. So the first thing is figuring out what is our stuff, what is our non-stuff, and then actually breaking it down into objects. I want to talk about the theory behind this first part at first, and then we're going to go through and do some demonstrations in CellProfiler on how this actually works in practice. The concept of thresholding is simple. It's just that when you have a digital image, which of course is just a bunch of different pixels that all have a different quantitative intensity value, you want to figure out which of those pixels belong to your stuff and which to your non-stuff. And if you're working with fluorescent images, essentially always what you're going to do to create a threshold is to look at the histogram of the values. So your background here in a typical fluorescent image might have a low pixel value, might not be that bright because there's nothing there. And your signal might actually live up here where there's higher pixel intensity values. And your goal in deciding where to draw your threshold is where to actually put it. If you put it too far to the left or too low of a threshold value, you're going to find your objects of interest, but you're also going to find a lot of background. That's obviously not ideal for a lot of applications. Likewise, if you go too far to the right and choose too high of a pixel value, you're going to make sure you don't include any background, but you might not find the object of interest. The goal with thresholding is to put yourself in this Goldilocks zone, to say, all right, what is the middle intensity value that allows me to find all of the pixels in the object that I care about and none of the pixels that I don't? Now that is what is going to be always the first step to getting your image analysis, and this is what we're going to do here in CellProfiler first to start. I'm going to start with a data set that we call advanced segmentation, and this is based on an actual cell painting data set. This is one of the example pipelines and example data sets you can find in our repo, our GitHub tutorials repo. Now this image set has 5 channels. It has a DAPI channel. It has a ER channel. It has a cyto channel, which stains RNA. It has a phalloidin channel, which also has WGA in it. And it has a mitochondrial channel. So we have 5 channels, all of the same. And the first thing you may notice as you look at these 5 channels is they all look pretty different. And if we go and we look at their histograms, this is the histogram for this image. Their histograms look pretty different too. So in CellProfiler, we offer a lot of different thresholding options, and which thresholding option you choose is going to be set by your image. Even in the exact same experiment, in the exact same field of view, based on the properties of our dyes, we have very different histograms, and we're therefore going to need to choose very different image analysis tools. I'm going to go ahead and close all these now. So I've already gone ahead and deleted most of the modules that were already in this pipeline because we want to develop a workshop here from scratch today. The one module I've left in is a flat field correction, where we want to actually go through and flat field correct out any changes in the illumination intensity from the image. I'm not going to show this for every field of view, but just to give you guys an idea why we might do this. So this is actually the illumination function of the microscope. We've calculated it in a previous pipeline. And you can see it's not the same all the way across the microscope. Down at the bottom, it's actually much brighter than up at the top. And so what this means in practice is that nuclei, or any objects that are found in the top of the image, are going to look artificially less bright than the ones at the bottom. So we can correct for this by dividing out the illumination correction. If you don't have to worry about this in your experiments, that's fine. This is just something that we find a lot of people do because microscope objectives, of course, are not completely flat. I'm going to close the eye on this here, though, because it's not what we care about right now. What we do care about right now is looking at the threshold for our different channels. Now, of course, the first thing for picking a thresholding algorithm is picking which channel you want to use, because as I showed you, the histograms for each of these images are really different. CellProfiler offers a lot of different thresholding methods, as you can see when I drop this down. I'm not really going to talk about the manual or measurement ones today, but you can set a manual threshold. We almost never think that that's a good idea. What we typically recommend you start with is something called the minimum cross entropy threshold, and this is just because it tends to work well in a broad variety of cases. I'm going to pick here an image set that I know has a lot of different kinds of thresholds in it, and I got here just by going to Test, Choose Image Set, and I can pick one that I've gone and looked ahead of time to know has a lot of different good thresholds we can play with. Go ahead and hit Run to run the two modules that are so far in my pipeline. This is what the minimum cross entropy threshold has come up with for this image. It has set a smoothing scale of 0, so it's not smoothing this image beforehand. And you can see that these numbers, final threshold and original threshold, are the same, and that's because our threshold correction factor is set to 1. The threshold correction factor is a setting that allows us to take whatever this original threshold is that's calculated and then multiply it times a number to make it either a little more strict or a little bit less strict. It's not something we recommend you necessarily do all the time. It's something you really need to go based on if you think your threshold consistently looks right or not. So if I zoom in on my nuclei here, I would say that this is doing a pretty good job of thresholding them. I like what it's picking out. I think it has the right pixels, and I don't think it has any of the wrong pixels. Another thresholding method that's very popular in the image analysis community is Otsu. Two-class Otsu is what we consider the standard. You can see it gives almost the exact same results here as minimum cross entropy. There are a couple differences. You can see here there are some dim areas in this nucleus that Otsu, which has a threshold of a little over 0.004, is not picking up, whereas minimum cross entropy, which said that a better threshold would be something more like 0.003, did pick up. But we're talking a tiny number of pixels across the whole image here. Otherwise, this is almost exactly the same threshold. So what you can hopefully also see is there are a range of values that might actually be correct for any given image, which is why, again, a manual threshold can be not a great thing to do. If I want to look at something with a little bit more complicated of a histogram, I can go back to that phalloidin and Golgi image that I was showing you earlier. Now, when I switch here to pH Golgi, which is what we've named this image here, what you can see is that two-class Otsu is doing a good job of finding something. It's finding, in general, the nuclei, which are sort of shaded in here, and it's picking up some of the background, but we're certainly not getting anything that's at all consistent. Now, if I remind you again what's in the histogram of this image, it looks like we have 3 peaks here. This is background peak, our cell cytoplasm peak, and our nuclear peak, most likely. Or it can also probably include some of this really bright stuff here. So if we wanted to find just the nuclei in this image, we could try actually a different kind of Otsu to see if we can bring out just that peak. With a 3-class Otsu, rather than drawing one dividing line between the 2 parts of your histogram, you actually start by drawing 2 to make 3 different peaks. You can only have one dividing line in your final thresholding solution, though. So this will ask you then if you want this middle peak, the one that's between the 2 lines, to count as positive, which is white down here, or negative, which is black down here. Let's start, if we said we want to find the nuclei, by assigning this to the background. So saying that this peak now is part of the background, we choose this dividing line here, and we only care about the top peak. If I do that, I do get something that approximates the nucleus. If I only had this channel and I needed to find nuclei, this is probably a good threshold to pick. We've got some junk that's around it and some bright spots that are coming from the other stains that are present here, but overall this is a reasonable approximation of the nuclei. If, on the other hand, I wanted to use my phalloidin signal to find all of the cell boundaries, since I do have a nice nuclear marker, I could say that I want to use this middle peak as foreground. I could set my ultimate dividing line here, and I could see if I can try and include all of these cytoplasm pixels in my threshold. And in fact, that's just what I see. By just changing this middle intensity class from foreground to background, I can get 2 totally different thresholds. With foreground, the middle intensity is part of the things that pass threshold, and we get a cytoplasm boundary. If it's background, then we get something that looks more like a nucleus. So hopefully you can start to see why we include so many of these different thresholding options. It's because everyone's images will look pretty different, and in fact, what you care about from image to image might actually look pretty different. Now, if I want to look at a different channel, I can try looking at the cyto channel, which was our RNA channel. Now, this threshold looks not that great at first. It's got a lot of stuff that doesn't look like, when I zoom in, is actually positive. Some of that's an artifact of just how these images are normalized and the fact that these bright dots inside the nucleus, which are the nucleoli, which are an RNA-rich region, are really bright. If I blow out the contrast, let's blow it out even more. You can see there is actually signal in all of the places that it's thresholded, and even some stuff up here that may be cytoplasm or may just be junk. But in any case, this is clearly not a thresholding method that's well matched to this channel, unless we wanted to use this channel to draw the cytoplasm boundaries, which it doesn't look like it's doing as good a job as the phalloidin channel because we have this extra crud that's hanging out up here. What we might want to do, though, is to use it to find these spots. Now, these spots are, like I said, are nucleoli, and they're definitely the brightest thing that's in this image. And if I ask to look at a histogram of this image, you can see here it looks pretty similar to our phalloidin one from before in that we have 3 peaks. So what will happen if instead of doing 3-class foreground, I try 3-class background here? What we have here is actually something that looks kind of like the nuclei. because it turned out our three peaks here are the background, that cytoplasmic signal we could see when we blew the image way out, and the nuclear signal. Our fourth class here, our fourth brightest class, these actual small little dots, actually don't even register as enough of a class for Atsu to see them. So if we wanted to find these nucleoli, how would we do it? Well, there's one more thresholding method here that we haven't tried yet. And that's called robust background. Now, robust background works a little bit differently in that what it tries to do is say, if there is a peak that's too small that you can't see, I'll just try to fit the background and then go a certain number of standard deviations above the mean. So let's open that histogram back up again. So what it will do is it will remove the top and the bottom of the distribution, so it will ignore all of these pixels, which contain our real signal, and some of the background. It'll try to figure out what is the mean of what's left and the standard deviation, and then go a certain number of standard deviations above the mean to say, all right, if we think the mean is probably something like this, the standard deviation might be something like 1 standard deviation, 2 standard deviations, and we might actually be able to push a threshold high enough where we get down into just this part of the tail and only our very, very brightest objects are visible. So that has gotten us a little bit higher. Before it was at 0.002, now it's at 0.0035. And in some of the cases, we are starting to actually see nucleoli come out, but it's definitely not bright enough. We've only gone 2 standard deviations above the mean, though. If we wanted, we could try to go more. Try and go 3 standard deviations. Okay, we're definitely doing better. 4. This looks pretty good in some areas, although now some of the dimmest nucleoli are dropping off. And we still have some areas where nucleoplasm is really bright. If we wanted to try to get rid of those, we could keep jumping distributions higher and higher. But eventually, we never lose this signal before we actually start losing real signal, like these dots here, which are gone now. So we can't pick up these nucleoli with a simple thresholding, at least not without getting some other stuff that we don't actually care about. But if we go back down to, say, 4, we could do a reasonable approximation of what these nuclei look like. And there are other image processing things we could do in a real pipeline to actually make sure that we figure out how to find just the nucleoli and nothing else. If you go to our tutorials repository and try out the advanced segmentation tutorial, it will teach you how to do exactly that. The one thing we haven't tried yet is setting thresholds in certain areas of the image. We've been setting what we call global thresholds, and I didn't explain what the difference between that and an adaptive threshold was for the ease of simplicity when we started. What an adaptive threshold means is that a different threshold can actually be calculated in different areas of the image. So a different threshold will be calculated over here as opposed to over here. And the goal there is to try to figure out, if you have illumination differences across your image, what is the brightest thing in a small window, even if the windows themselves are different because there's lighting conditions that are different across your image. There are other ways like background subtraction to try to get rid of those, but this is one other option. Only Atsu thresholding is available in the adaptive mode, at least in CellProfiler 3. But we have all of the same Atsu options that we had before. Now, Atsu 3 class with the middle class to the background, aka pull out only the brightest of the 3 peaks, was the thing that was closest before. So I can give that a try and see if it will actually help me get a better threshold than what I have so far with robust background. And with a window size of 50 pixels, aka it tries to find what is the brightest thing in this window, I'm still basically picking up just nuclei. I can try a smaller window, so I can try and get some windows that include just these dots without a lot of background. And in some places now, it is actually giving me the nucleoli, but it's definitely not working across the board. And in some areas where my 10 pixel window doesn't include any nuclei or any nucleoli, it's actually just picking up the cytoplasm and calling it thresholded positive. So this isn't a good option here, but it can be a good option in some other circumstances. There's no way with a simple thresholding we can actually pull out the nucleoli in this pipeline. Ultimately, though, we're not going to use just thresholding to find objects. Remember, I said object identification is a two-stage process. It's very important to figure out what is stuff and what isn't stuff, and to get your threshold right. In fact, everything else that you do in CellProfiler is going to follow downstream from that. But it's also extremely important that once you have found the threshold and found what parts of your image are signal or not signal, that you actually are able to go through and identify and break things into objects. We don't just want to know that this central area of this image is positive. We want to know that there are 1, 2, 3, 4, 5, 6, 7, 8 cells in it. And there are a couple different ways that CellProfiler can do that. Let's go now into looking at actual object detection in CellProfiler. Now, in general, you're going to start with the Identify Primary Objects module. Identify Primary Objects is used whenever you want to find objects in an image and you don't have any previous objects that should be taken into account. You'll see in a little bit exactly what I mean by that. But if I want to start by identifying the nuclei, I can go into test mode. I can find my good test image. And when I add Identify Primary Objects to my CellProfiler pipeline, I don't have a ton of options I can configure because I have advanced settings set to no. I'm going to start just with the most basic settings that I have. Okay, this is CellProfiler's first pass at doing a segmentation. So now not only are we determining which pixels are positive and which pixels are negative, CellProfiler is trying to break them down. We can't see yet how it's trying to do that, we just know that it is. If I zoom in, I can see that my threshold still looks pretty good here. It looks like it's doing a good job of the first step, which is figuring out what is stuff or non-stuff. What it's not doing a good job of is breaking stuff down into objects. Now, the only tunable thing I have at this stage is what size it thinks my objects should be. So the default here was 10 to 40, and I didn't change that at all. Is that actually the right size? If I go to Tools, Measure Length, I can try and get some diameters of these nuclei to see about how big they are across by clicking and dragging. That one is 53.8 across, 28.6, 38.1. So some of them would fit in 10 to 40, but definitely a lot of them don't. What if I tried instead something like 20 to 100? Okay, that looks a lot better. It's still not quite breaking this one in quite the right place, but overall, this is a pretty good job. We have two things that are broken where they shouldn't be, but overall I can live with that. If I want to see how this now looks across a few other image sets, I can just go to Test Random Image Set and have CellProfiler feed me a few more random images to make sure these settings are good across the board. This is okay. The threshold is not great. or is great, but some of the places where this is being broken are not quite right. We want to see how this looks on a few different images though. This one looks pretty good. That one looks really good. And so does that one. So overall, these settings are pretty good. We don't want to change them too much, but there are some places here where stuff doesn't look like it's being split quite right. So I'm going to go ahead and pick out one that I know needs some extra work. Now you can see here there are a lot of things that are being reflected that I haven't explained yet, because we haven't really gone into the advanced details of how CellProfiler is breaking these into objects. We wanted to just, as a first pass, see if telling it how big the objects is good enough. And it is almost good enough, but let's go further into the details now. If I turn on advanced settings, I get a lot more options. These discard images, objects outside the diameter range and touching the border of the image were there before, and they're what are making our pink and yellow objects. Pink objects are things that are too small, and yellow things are being thrown out for touching the border. I could set both of those to no if I wanted, but then I might include some things that are too small that aren't really nuclei, and I might find some stuff that's just half a nucleus. In fact, you can see if I zoom way in here, just a little piece of crud at the edge of the image. It might be a nucleus, it might not, but it's certainly not something I want to include. I'm going to go ahead and set those back to yes. The first several settings that you see should look really familiar because they're the settings of the threshold module, and in fact they are exactly the same as the settings in the threshold module, with the exception of that this image gets now smoothed a little bit before we draw the boundaries. This just typically helps with making nice smooth outlines of objects. If you wanted, you could set it to a much larger smoothing, but some of the boundaries now start to get pretty blurry, and you can see here, for example, that now the boundaries of this have pushed way out where they really shouldn't because I've smoothed this image quite a lot before thresholding it. So I'm going to put it back where it was. The threshold correction factor is just what it was before. But now we have a bunch of options for how we actually draw dividing lines between objects. Now, what does that actually mean? Splitting thresholded images into objects is a two-step process in CellProfiler. The first step is figuring out where the centers of the objects are, and it can do this based in two ways. It can do it based on intensity. It can say what is the brightest area that I can find, and I will set that as a high intensity pixel, and I will think that that's the center of an object, because often in a fluorescence image, like this one we have before, which is our same picture of cells, but now the brightness of the intensity is being visualized as height. Very often in a fluorescent image, the center of an object is the brightest point. The other way we can do this is if we know our objects are roughly round or blob-like, we can say that we want to break this into roughly round or blob-like options by saying the center are the points that are furthest from any edge. Now that is true in a circle. The furthest point from any edge is the center of the circle. So if we say, take the furthest point from any edge as our object center for a relatively round object, that'll work great. And that'll mean that because here we have an indentation, this point is not very far from edges. This point is very far from edges, so the actual object centers will get defined here and here. Once we find the centers, we can then choose to either draw boundary lines around each center based on either a dip in the intensity, just like we see here. We could essentially follow where these mountains hit the ground, or we can use shape again to follow boundary lines to make things into approximately round images. Now, which one will work best is sometimes obvious. If you want to study actin filaments, trying to break things into round objects isn't going to be a very good idea. But when you're looking at something like nuclei, either option might be good, so we should try both. And the default that was set before, that was hidden from us when we had advanced settings hidden, was intensity and intensity. Now, for the most part, that isn't doing a bad job, but we have some things that are stuck together up here and down here. Now, these nuclei look pretty round to me, so what happens if I just tell it that I want to break it down into shape instead? I'm doing a better job down here now. This nucleus, which was before being seen as 2, is now being broken in half. And this clump up here that was not being broken up before is now correctly being broken up. We're still not quite perfect though, because we now have this big clump here that when I was going by intensity was correctly being broken into 3 nuclei. So we're in a case where either one will give you approximately the right answer, and the differences between them are small. But in your image, it might be very different. The one thing that we have over here is this big clump of nuclei, which I'm going to choose to allow the program to just keep all together so that they're thrown away, because these are hard to segment. But if it were important to you to actually be able to get every nucleus, of course you would want to try to fix this. How would you fix it? Now, there are a couple settings here that are related to how CellProfiler, when it actually does that finding of centers, actually does some things that I didn't explain to you before. If we zoom in here, you'll see that this nucleus here has several actual small bright spots within it. Any one of these could have been found as the center of this nucleus. But CellProfiler, based on the size of the objects that we told it to expect, did 2 things without telling us. The first thing that it did is it just smoothed this image after it was thresholded, but before it did the declumping, to try to figure out, to try to smooth out small imperfections like this. Now you can see this is a much bigger smoothing filter than the one at the thresholding level. That's okay because we know what is signal and what is not signal. We're just trying to break things into bits. The other thing it did is what it calls maxima suppression. So based on how big you tell it that the object should be, if it finds 2 bright spots that are very close together, if they're just closer together than this maxima suppression size, it says, oh, I think those are 2 bright dots in one object and not 2 different object centers. I'll ignore all but one of them. So this chain of 4 bright spots across the center of this is all within a pretty close distance to each other. So the maxima suppression size only allowed it to have one of these bright dots, and we don't know which one, but it doesn't really matter for our purposes, to be called the center of an object and ignored the rest of them. Now overall, I think this is doing a good job of splitting things to be about the right nucleus size. But as a demonstration, what I can do is I can show you what changing those settings looks like. And I do that by just setting these things which were set to automatically calculate before, which you might not even have noticed. Now I want to manually calculate them. So the values for both of these started at about 14. If I try cutting them both in half and going to 7 for each, what you can see here now is that these objects have been broken up a lot more. They're broken into a lot more pieces. Some of them are still actually being all called together, but some of them have now been broken up pretty small, probably smaller than we want. For example, here's a nucleus that has a little Pac-Man chunk cut out of it. If I go to the opposite end of the spectrum and make each of these twice as big as they were in our initial automatic settings, now everything is stuck together, as we can tell pretty easily by the colors here. For example, this chunk group of nuclei is now all one big chunk, one big nucleus, which obviously isn't correct. There are 1, 2, 3, 4 here. So if I go back to the automatic settings, CellProfiler in this case is doing a pretty good job. And these automatic values are based on the lower end of this typical diameter range. If CellProfiler isn't doing a good job in your image, you can make these values either slightly bigger or slightly smaller than the automatically determined ones to try to adjust to what degree things are being split or merged. I will point out to you, though, that even when we changed the settings a lot, sometimes a given nucleus isn't going to get split the way you want it. And in general, an image analysis pipeline is never going to return 100% the values you want. So when you get to a point where almost all of the image is right, and you have a few things that are being clumped together incorrectly and a few things that are being broken incorrectly, that's usually when I say that it's about time to call it a day. And I think these nuclear settings are actually good enough. Though let's double check now on another image. So anytime we make a change here, we want to make sure that it looks good on other images, not just on one. Okay, I think we're in good shape. Now, CellProfiler also has the concept of a secondary object. A secondary object involves taking a primary object, such as a nucleus, and building something around it, like a cell body. Now, this isn't something that you need to do in every experiment, but if you start by identifying nuclei, which is typically a lot easier than identifying cells themselves, this gives you a really powerful option to be able to make sure in a mononucleate cell line, or any case where each one cell does correspond to one nucleus, you can make your identification process a lot more smooth by starting from the nuclei and building around them. So before we looked at the actin image, and that looked like it was pretty good for figuring out where the cell boundaries were. So if I want to identify something that I'm going to call cells, I'm going to start from my nuclei, and I'm going to build out around them. And again, the first thing that we're going to do, just like with primary objects, is to run a threshold. So unfortunately, this threshold doesn't look like it's doing a very good job. It doesn't look like it's nicely expanding out around these nuclei. Let's try a few more to see if this is a problem just for this image or if this is a problem sort of across the board. Okay, the threshold looks pretty bad here too, and it's about 0.006. That threshold looks a lot better, and it's about 0.004. So the pattern I'm noticing as we go through here is that when we have nuclei that are well separated, and therefore we have some background pixels in our cytoplasm because our cytoplasm is well spaced, we do an okay job of finding the cell boundaries. In cases where the nuclei are pretty packed together and there isn't a lot of background in our image, then the cytoplasmic segmentation is not doing a very good job of finding the edges. And the big part of that is because it expects that the histogram is going to have a foreground and a background peak, and we don't really have any background present in those images. Here's a good example of one that I think is doing a pretty good job. This is picking a threshold of about 0.003. Our background is pretty consistently at 0.002. Now the problem here, like I said, is that there are no background pixels. So one thing that we can do is we can allow CellProfiler to choose the threshold under certain circumstances, but put some boundaries on it so that in cases where the thresholding algorithm simply can't do a very good job, we override its decisions. And the settings for doing this are found here, the lower and upper bounds on the threshold. We return back to our histogram, and we go back to our idea of that we're simply trying to find the best threshold that divides the intensity. We can say, based on our knowledge of the images, that there are reasonable lower and upper bounds of what that threshold can fall in. On this histogram, for example, we know that the threshold for determining signal from not signal is probably not here. It's probably not over here. It's probably somewhere in this. And so if we look at all of our images are very consistent, and we have a reasonable set of data that we know supports this, we can decide to only let thresholds fall in that region. What this looks like in practice is that CellProfiler will attempt to draw a threshold on every image based on its pixel values. And if that intensity threshold falls within the lower and upper bound, CellProfiler accepts that threshold and then uses it to figure out what is positive and what is negative in the image. If any other threshold falls inside that value, we still accept it. But if CellProfiler decides to put in a threshold based on the pixels that are present that is outside of our bounds, in this case, it's too low, but it would also do the same thing if it were too high. It gets rid of that threshold and sets it to the closest bound. So in this case, it's now ignored that this threshold was too low and set it here. Now, none of the threshold algorithms we have will work nicely in Identify Secondary Objects for cases where the entire image is confluent. So this one that looks like it's 0.003 looks good. But this one that has a value of 0.005 and some of the other ones that I noted had values of about 0.005 are bad. And if I hover my mouse over the cytoplasm values, I see that they typically do have values that are about 0.004 or 0.005, some of them about to 0.0035. So what I can do here is I can set an upper bound and I can say that CellProfiler can never allow a threshold that's higher than that. Now again, this is something you want to do only when you need to, because in general, you want CellProfiler to be able to adapt to the robustness that's in your images. But when you consistently run into problems that can't be solved by any other solution, setting boundaries allows you to keep the threshold within a certain regime. Now, when I look at my nuclei and cytoplasmic thresholds, even in cases where there's very little background and there's a lot of signal, it stays within a value that makes the actual cytoplasm look good here. Now, in terms of where it draws the dividing lines, we have a few different options here. Propagation is the most commonly used one, And what that will do is it will try to balance out to make all of your objects about the same size. That's often helpful because cell boundaries can be really tricky to divide. But if you don't want to do that, you can switch to something like one of our watershed options, which will very strictly try to follow the intensity gradients that are present in the image. So you're going to get something that looks like a more realistic cell boundary, and in some cases it will be a better cell boundary, but in some cases it might be worse. Watershed image looks like it's doing okay here, but it's definitely making some mistakes in some places. And so we now have some cells that are being identified as tiny and some cells that are being identified as huge. We can also use distance thresholding to essentially, around our nucleus, draw the cell boundary just by expanding out in a sort of donut-like fashion. So I could say that I want these to be 50 pixel donuts around my nucleus. Now, you can tell that in some cases it has to stop long before it gets to 50 pixels because the cells just aren't that large. What you can also see is that with distance n, it's going to expand out into even some areas that are background. Now, that might be good if you, say, don't have a cell boundary marker and you literally just want to make the cells an exactly even size around all of your nuclei. You can also split the difference and use what we call distance b, which will try to make those donuts and will make those donuts unless it falls into a background area, in which case overall we've made these sort of donut shapes around the cells, but even when the edge of the cell is not 50 pixels away from the background, it stops at the background edge. In the last 10 minutes, I want to return back to our identify primary objects, our major kind of segmentation. But I want to talk about it now in the context of 3D. In 2017, CellProfiler released CellProfiler 3.0, which allowed you to do 3D segmentations. Close these guys. We have a tutorial here called 3D monolayer that's designed for use with 3D images. I want to load that up here for you to show you the difference between 2D and 3D. Put it in my pipeline. Load in my images. The first thing you'll notice is that the CellProfiler display windows look pretty different in 3D than they did in 2D. We're now seeing 9 slices taken through the Z-stack. We're not seeing one image at a time. But what you also probably notice about this pipeline is there is absolutely no Identify Primary Objects module in sight. And that's because Identify Primary Objects doesn't support 3D. Now, there's a reason that we've decided not to allow it to do that, and that's because in 3D, a lot of these operations are slower. We've walked through Identify Primary Objects, and it's taken me half an hour to tell you all of the different things that Identify Primary Objects is doing. And it does them very quickly in 2D. You hit run or step, and within a couple fractions of a second, you get an answer out. But that isn't the case in 3D. So in 3D, all of the same steps about segmentation are true, and we'll go through them now, but they're all broken out individually. But if you remember that what we do in 3D matches what we do in 2D, nothing we do here will be a surprise, and you'll know how to put things together. This starts just with some rescale intensity modules, just to make things equally bright. You don't need to worry about those right now. The first step that we have here is just resizing to make this run a little faster so that I can show this to you in a timely fashion. So we're just making our nuclei about half as big. And the next step is doing a filtering. In exactly the same way we have this threshold smoothing scale where I said we smooth the image before we threshold it. This smoothing option here is performing the same function. We're using a median filter here for smoothing. but you could also use something like a Gaussian. We then do a threshold in the same way we do in identify primary objects. And we get something that looks pretty similar to what I showed you guys in 2D when we were looking just at the thresholding steps in 2D. The next step is remove holes, which CellProfiler is actually also doing in 2D. It's deciding whether or not you want to fill holes after thresholding or not. Often you want to do that. Not always, but often. There aren't a ton of holes here, but it has filled in, for example, some of the gaps here. And once it's done that thresholding, it does a watershed, which is the breaking down. And it can do that only in what is equivalent to CellProfiler's shape-based mode here, which is called distance. But this is the same mathematics that was underlying CellProfiler's shape-based declumping. You can see here, overall, we have a couple segmentation mistakes right here in the middle of this nucleus, but for the most part, it's doing a good job of breaking these into spheres. And because we did this as a half-size thing just for processing time, we now just have to make our nuclei twice as big. Now, if we want, we can, in the same way that CellProfiler filled in holes in the objects after declumping, we can do the same thing in 3D as well. But again, this is an option, just like it is in CellProfiler. We don't actually need to do that. Now, if we want, because in this particular 3D example, we don't have a cytoplasm channel, this now goes through some steps to try to create essentially a cytoplasm mask that is similar to our 3D. It does that by taking a membrane channel, inverting it, and trying to fill some holes in it until we end up with something that looks like this, which is the equivalent of our thresholded phalloidin channel over here, just made by inverting the membrane channel. And in the same way, we use identify secondary objects in 2D. We can start from a central object, which over here is just called a marker, go around the nuclei, and make objects in 3D. So we can follow that same, find the easiest object to find, and then expand it out to the edge of the boundaries in 2D or in 3D. The names of the modules are a little bit different, but the theory behind them is the same. I hope that's helped you understand a little bit more the objects that we have present, the options that we have present in CellProfiler for finding objects, and how you might change each of the different settings in order to find the things that you care about. It takes some work to try to go through, test on a bunch of images, and make sure that whatever settings you pick apply well across the board. You might need to put some boundaries on things, and you might need to try a few different thresholding options or a few different declumping options before you're happy. But overall, you should be able to find the objects you care about, generate measurements, and do good reproducible science based on that. Thank you guys for paying attention. I hope you have a good rest of your day and your weekend. Stay safe.